What is gravimetric analysis? Gravimetric analysis is a chemical technique for determining the mass of a substance. The approach is based on the idea that every material has a mass that can be measured. In this method, a known mass of the chemical is weighed and then transferred to a vessel. The material is then evaporated by heating the vessel.
A quantitative analytical method called gravimetric analysis uses a substance mass to determine how much of it is in a sample. Gravimetric analysis is a widely used technique in pharmacy to determine the concentration of certain ingredient in a pharmaceutical sample. Many fundamental ideas form the theoretical foundation of pharmaceutical gravimetric analysis. Number 1. Formation of a precipitate. Gravimetric analysis often relies on the formation of a solid, insoluble precipitate through a chemical reaction between the analyte and a suitable reagent. The reaction should be selective, ensuring that only the analyte of interest react to form the precipitate. Number 2. Stoichiometry of the reaction. The stoichiometry of the reaction between the analyte and the precipitating reagent should be well defined. This principle is important for accurately relating the mass of the precipitate to the amount of analyte in the sample. Number 3. Chemical purity of reagents. High purity reagents are required for gravimetric analysis. Reagent impurities may cause the precipitate to become contaminated and produce unreliable findings. Number 4. Ignition or heating step. The precipitate may occasionally be heated to a consistent mass following filtration. In order to guarantee that only the intended substances contributes to the mass measurement, this procedure eliminates water or volatile components. Number 5. Quantitative precipitation. All of the analytes should react to generate the precipitate in a quantitative precipitation reaction. Errors in the analysis may arise from any incomplete or unintended reaction. Number 6. Isolation of the precipitate. It must be simple to separate the precipitate that has developed from the solution. To separate and purify the precipitate, effective filtration and washing methods are used.
Next is methodology. There are 5 steps involved. The first step is preliminary treatment, which is the preparation of the solution that contains sample. The purpose of this step is to adjust the solution condition before precipitation. The second step is precipitation of precipitate. The precipitating agent should react with the analyte to give precipitate that consists of large crystals that can be easily filtered and free of contaminants. Insoluble, so the amount of analyte lost during filtration and washing will be really low. Stable, so the precipitate unreactive with constituent atmosphere and non-composition after it is dry. The third step is digestion. Digestion is the process keeping the precipitate within its mother liquid for a certain period of time to encourage densification of nuclei. This process helps produce larger crystals that are more easily filtered from the solution. Next is filtration and washing. The purpose of filtration process is to separate the precipitate from the supernatant solution and other constituents of the sample, while washing is required because precipitating reagent is usually added in excess. The last step is drying and ignition. After filtering, the gravimetric precipitate is heated until its weight becomes constant. Heating removes the solvent, absorbs electrolyte and any volatile species that is carried down with the precipitate. Some precipitate must be emitted to decompose the solid and form a compound of stable and known composition.
Example of gravimetric analysis in pharmacy.
For example 1, tablet friability testing. Tablets are weighed before and after subjecting them to mechanical stress in a friability. The mass loss determines tablet friability and durability. Second, pre-formulation studies. During early drug development, gravimetric techniques can evaluate various solid state properties like density, flowability, and surface area of drug powders that impact formulation development. Validation of cleaning procedures. Gravimetric method can quantify drug residue level of manufacturing equipment. After cleaning to validate that cleaning procedures effectively remove residues below permissible exposure limits. Other than that, gravimetric analysis also used in analysis of creams and ointments. The water content in topical formulation like creams and ointments can be determined through loss on drying methods. This helps assess product stability and antimicrobial preservation. Gravimetric analysis, it is used to measure the volume of plasma. Radioiodinated human serum, albumin, and red blood cells can be utilized in the same way to calculate plasma volume. In fact, this is one of the most widely used applications of gravimetry in pharmacy. Lastly, calculation of encapsulation efficiency. In products like microspheres, nanoparticles, and liposomes, the encapsulated drug is separated from the free and untrapped drug, and each is quantified gravimetrically to determine percent encapsulation efficiency.
Gravimetric analysis in pharmacy is a methodical technique that starts with sample preparation and ends with calculation based on reaction stoichiometry. It forms an exact precipitate through a quantitative reaction to precisely determine analyte mass. Pharmacy quality control, impurity identification, and medication assay all benefit from the flexibility and dependability of gravimetric analysis. Its accuracy depends on employing high-purity reagents, adhering to established protocols, and paying close attention to experimental details. Even with the improvements in the pharmaceutical business, gravimetric analysis is still an essential tool for ensuring the quality and efficacy of pharmaceuticals. It is a dependable method for quantitative determination in formulation, manufacturing, and quality assurance.